My name is Tanya Dyer, and I'm from Bermuda. I have an 18-year-old son who has arterial tortuosity syndrome.
The disorder is so rare, and there are only a few patients even in the world. We could never dream of it because it's so rare, right?
But with the nonprofit and all the other mothers behind me, I didn't feel alone anymore. And I could see that Tyrese, once he met those kids at the Arkansas conference, he didn't feel alone anymore either.
The more samples we have, the more research we can conduct, and it also can account for the variability that's seen in patients. It will be easier for us to make conclusions from the data we have if we have more samples.
And they had called in a geneticist, Dr. Stephen Kaler, who came in and spent the whole evening with us asking us questions and going back and doing research and back and forth. And by the end of that night, he came back and said, I think I've got it. I think Aiden has what they call arterial tortuosity syndrome.
I met Aiden in 2010. At the time, we didn't know his diagnosis. He went to the cardiac catheterization laboratory because of concerns for pulmonary hypertension. And during the procedure, we found a very unusual tortuosity of his vessels involving his lungs and his great arteries to his head and neck. And following that catheterization, I inquired from one of my genetic colleagues about what could possibly be the cause of this picture on the angiogram, and that's how we stumbled onto the diagnosis of ATS.
As a parent, you never want to hear, I don't know what ATS is. You never want your child treated just like every other child to fit in this nice, neat little package. Arterial tortuosity syndrome is very unique, and each individual child with ATS will have their own symptoms and react to different procedures in their own special way. So if the physician tries to treat a kid with ATS in the box, they're gonna mess everything up.
We're very interested to work on it because it's rare and those patients are affected, and you want to, it's a kind of passion to find something and to get the answer on many questions.
So my name is Bert Kallewaert. I'm a clinical geneticist, head of clinic at the Center for Medical Genetics in Ghent. That's in Belgium. I'm associate professor at Ghent University. I'm a pediatrician from training, but I am actually working as a clinical geneticist.
My name is Paul Koeken, and I'm a staff member at the genetic department here in Ghent, and my profession is I'm a molecular geneticist who is responsible for the connective tissue lab. In 2003, when my promoter at that time came to the attention or said that there was an interesting family with ATS, a Moroccan family, and he was able to get some DNA samples of that family with 7 or 8 affected family members. And at that time, in 2003, the gene for ATS was not known. And it was possible to perform linkage analysis with his family to identify the ATS gene. Linkage analysis is a way to identify a gene, and this is only possible when you have a large family with several affected family members, but also with family members who do not have the disease, right? And so you compare pieces of DNA of the affected members with the non-affected members, and all the affected members should have pieces of DNA which are not present in the non-affected members. And with linkage analysis, you try to identify those pieces of DNA, so which are very specific, present in the affected family members. Identifying new genes was very important, and was great in science, but also for the patients, because this was the first step for them to have a diagnosis, a molecular diagnosis, but also especially for the parents, if they would like to avoid a second child in the future, they could do with this kind of information prenatal analysis. And it's also the first step in the identification of the gene. Yeah? First you have to know where, on which chromosome it's located. And once you know this, you can identify on that chromosome the gene. And at that time, this was the first step we had. The second step we had to make was to identify the gene there. And we knew that in that region of the chromosome, approximately 50 genes were located. So we were looking to the most relevant candidate genes in that region. And it was the work of the people as Professor Callewaert and Andy Willard, Dr. Willard, who were working on this very specifically and very focused, and they started with the most relevant genes, which are structural genes. Because of arterial tortuosity, you think arteries are affected because there is a structural gene defect. So they selected structural genes. And after 49 genes of the 50, they didn't find anything. After months and even years of analyzing and checking genes. And then they took the last gene, which was a glucose transporter, which was not at all a candidate for arterial tortuosity, of course. And then they found mutations in that specific gene. And this was so unexpected that even Nature Genetics, it came to the attention of Nature Genetics, and they were very much interested because of the unexpected issue or the unexpected finding that a glucose transporter, which was thought at that time, was responsible for a structural defect or a structural disorder.
Actually, when I started my PhD, Paul was one of my supervisors. And so he already had the region on the chromosome where the gene was expected to reside. And so I started screening all those genes, which was indeed a very difficult task because it was quite unexpected to find mutations in that specific GLUT10 gene. And so that actually caught my interest. Meanwhile, I was doing clinics on connective tissue disorders in general. And so I got in touch with patients with ATS, but also with other connective tissue disorders. And that actually caught my interest because these patients are facing amazing problems, and we don't have answers yet to those problems. So that's something of our goals that have been set forth, that we want to find better treatments, better management guidelines in order to help those patients.
So when you send the tissue, we can do a lot of different things with it. It depends on how the tissue is sent to us. We can now do three things. If the tissue is in culture medium, we can use the tissue to cultivate fibroblast cells from your skin biopsy. It's actually the same for a pulmonary artery biopsy or an aortic biopsy. If your skin is in another fluid, a fixation fluid, we can actually embed it in paraffin, make small sections, and really look at the histology of the tissue. And if your skin biopsy is in an other fixative, we can use it for electron microscopy and really look at the tiniest structures of the cell.
Research is also important because we want to know the exact mechanisms between the gene defect and why it's giving those clinical features of arterial tortuosity and arterial aneurysms. And so that is something that is often difficult just to do on either blood samples or some biopsies. It may help, but one of the most important things is to have a valuable animal model for that.
So I'm not going to touch them because they're just newly born. These were born probably yesterday, so these are 1 day old probably. And the mom's protecting them.
I didn't see the authorization. So are these the SLC2A10 mice, or are they just random mice?
Yes, these are the mice we are doing the actual research on. So
Look at all the babies.
the
So is
eyes
that
you
two
saw
mamas,
earlier,
or is it
it's the mother and the father normally. I can show you.
And so what we initially started with is making a mouse model, an existing mice model. However, it turned out that the mice didn't show any particular phenotype that could be linked to ATS. There was no tortuosity, there were no stenosis, there were no aneurysm formations. So we had to take another approach. So the next level we will go to is actually making a zebrafish model and see if those fish may actually show clear anomalies that can be related to ATS.
She's collecting the germ cells of the fish, and those were actually fish that she injected with CRISPR-Cas components. And those components, they can mutate a certain gene of interest. So it's just the same that I did with the ATS gene, with SLC2A10. When we want to mutate a fish, we inject the mutating components in the embryo when it's small. And then when the fish is adult, we collect the germ cells. Like this. We collect them separately, and then we store the fish also separately until we have analyzed all the results. And in the germ cells, we are looking for the specific mutations in the gene of our interest. And if we found one, then the fish is positive, and we will use it for further breeding.
We have access to all kinds of tissues: arterial tissue, heart tissue, skin tissue, muscle tissue, and so forth. So this would be truly valuable to study the mechanisms in detail and find the exact connection between the gene defect and the clinical presentation. And having those connections, this will lead to opportunities to try and do some compound screening in order to find medications that might be helpful.
Because of the unexpected finding, we were able to publish this in a high-ranked journal, this information. Of course, which was very important that at that time, from now on, we could detect and identify and make a molecular diagnosis in those patients, which is very important because of the clinical spectrum, which is not easy to differentiate with other kind of disorders. So for those patients, it's important to know exactly what kind of disease or which molecular diagnosis they have. And it was also important for the patients in this finding. Right? There are only a few labs in Europe who are working on connective tissue disorders. Especially 10-15 years ago, there was only 1 or 2 labs who were working on this kind of disorders, because actually those connective tissue diseases, many large genes are responsible, right? You have fibrillin 1 and the collagen 5 and the collagen 3, and these are huge genes. And nobody was interested in the past to work on huge genes because it was too difficult. It took too much time to analyze them. Yeah? So we got samples from Spain, from Italy, from Poland, from Germany, because nobody liked to work on this kind of connective tissue disorders. And so there were only 1 or 2 or 3 labs in Europe. And of course now it changed because sequencing became much cheaper and it's much easier. Although we have the experience from 20 years to work on this kind of disorder, so we are ahead on other ones who just started with it. So I think Ghent is an important setting to work on this kind of disorders. Especially also as we have the clinicians involved. Our lab has the luxury that although there are molecular geneticists and there are molecular biologists, there are also a great team of clinicians, and we have the connection between both, which is not always the case in research labs, for example, where they don't have the patient material or they don't have the connection with the patients. So therefore Ghent is a very important setting to do this kind of research.
All the skin samples and the cell cultures, when we don't keep them in culture, we store them in liquid nitrogen. That's at minus 160, minus 180 degrees. And in this way we can store them for a very long time. And when we need the cells again, we get them out and bring them again in culture. It's a process that we can repeat many times. And then we store the cells in racks like this. So in each tube there are the cells of some patients on which we do research.
I'm Udis Fontenot. I'm a pediatric cardiologist. I'm a professor of pediatrics at the University of Arkansas for Medical Sciences, and I'm based at Arkansas Children's Hospital.
Hi, my name is Aiden. I am 9 years old and I have ATS. My name is Megan and I'm 6 years old.
ATS is a genetic abnormality that produces abnormal growth in certain vessels, and the vessels, because of their abnormal growth, become long and curved and bendy, and so hence the term tortuosity. So it's a genetic abnormality having to do with abnormal vessel growth.
ATS makes me have to go to a doctor and to go to the hospital a lot. ATS causes your arteries to grow too long and can causing you to have lots of surgery.
The symptoms from a child with ATS can be varied. It's sometimes very difficult to make the diagnosis because the presentation in the children, the way they present and the organs that are involved seem to be different from one child to the other. So depending on where the vessels are that are involved in the syndrome will be the presentation. Some children present with abnormal vessels in their lungs, some present with abnormal vessels in the head and neck arteries, in the brain, and the other body organs. So there's lots of, there's a varied presentation because of the abnormalities associated with the disease. And we're still discovering and describing and learning these presentations so we can be more adept at making the diagnosis when these children present.
My name is Andrea Taylor, and I have 2 boys, Aaron and Aiden, and we are from Arkansas, but currently live in Oklahoma. Our story with arterial tortuosity syndrome really started with the birth of Aiden in 2008. The pregnancy was normal. We didn't have any kind of complications, no gestational diabetes or elevated blood pressures or anything like that. When he was born, everything seemed fine. You know, we noticed a few little things here and there about Aiden, that he had a very long torso and that he had a little bit of an aged appearance. We joked that he reminded us of my grandfather a lot.
My name is Ginger Knapp, Ginger, Knapp, Knapp. I'm from Wisconsin. I live there with my family, my husband and 3 children. Our youngest daughter was diagnosed with ATS almost 3 years ago now. Her story really began at birth, where there was symptoms like problems with her hip, problems with her muscle tone, some gross motor delays. She ultimately ended up getting picked up by genetics, and she has a very unique story because she had some findings that were really not previously reported in medical literature.
My name is Tanya Dyer, and I'm from Bermuda. I have an 18-year-old son who has arterial tortuosity syndrome. I had a normal pregnancy, and Tyrese was normal. However, at 6 weeks old, he started projectile vomiting and he had to have a pyloric stenosis repair. So we went back to the hospital and he had to be air ambulanced to Boston Children's Hospital.
In the spring of 2010, Aiden had 2 pneumonia-type illnesses that required hospitalization about 6 weeks apart. After they did several rounds of different tests, they decided that Aiden needed to go in the cath lab and see if they could relieve some of the pressures on his heart, because his heart had already started to enlarge.
I met Aiden in 2010. At the time, we didn't know his diagnosis. He went to the cardiac catheterization laboratory because of concerns for pulmonary hypertension. And during the procedure, we found a very unusual tortuosity of his vessels involving his lungs and his great arteries to his head and neck. And following that catheterization, I inquired from one of my genetic colleagues about what could possibly be the cause of this picture on the angiogram, and that's how we stumbled onto the diagnosis of ATS.
When he went into the cath, we knew that there was a possibility that it was exploratory and they didn't know if he would make it out of the cath. They didn't know what to expect. Aiden was really sick at this point. When Dr. Fontenot came out of the cath lab, his head was down, he was very distraught looking, and you could tell that he really didn't know how to talk to us. And so he began telling us about the tortuous vessels and how he could only go so far, and how they had everyone looking at his images, and they had called in a geneticist, Dr. Stephen Kaler, who came in and spent the whole evening with us asking us questions and going back and doing research. And by the end of that night, he came back and said, "I think I've got it. I think Aiden has what they call arterial tortuosity syndrome."
So because some of those findings had not been previously reported, it made her a little bit more difficult to diagnose, because those pieces that hadn't been previously recorded in medical literature weren't fitting in to the typical profiles of the diseases and the symptoms she was having. So ultimately she did have whole exome sequencing, which did confirm the diagnosis of arterial tortuosity syndrome. One time, when I was specifically talking about some of her joints and stability of her joints, I had mentioned to one of the doctors, I'm like, boy, if it's this bad now, I think I started off by saying, well, how would you rate this? He was like, severe. And I said, well, if it's severe now, how do we predict that? And he said, I won't answer that. He said, I just don't know.
My daughter was diagnosed with ATS when she was around 3. We took a trip to Mayo Clinic, and that's when she was diagnosed with ATS.
I pretty much wake up in the morning thinking about ATS, and I go to bed at night thinking about ATS. ATS somewhat rules our life. At what point am I going to get the news that we need to have a surgery or go to the cath lab? Because she's been lucky that Maggie has not had to go to the cath lab yet. But with every doctor appointment, it sort of makes you wonder, is this going to be the day that we're going to get some sort of really bad news?
Sometimes the kids get pushed off to the side when there's too much going on with Maggie, and we have to remember that our kids need attention separately.
So now things have changed in the sense of, now that we know that is her disease name, and she has the diagnostics to sort of back that up, the pieces that hadn't been previously recorded are, I think, opening a lot of new doors to different research.
So they repaired the stomach hernia and they did a fundoplication, which would keep the stomach from moving upwards. I was told later by his pediatrician that if they had waited any longer in Bermuda, the stomach would have collapsed his lungs and he would have died. A year later, we had to go back to Boston for a follow-up. When we got there in November, they detected a murmur. When Tyrese was about 4 years old, they clinically diagnosed him with Ehlers-Danlos, which was a connective tissue disorder. Every time I went back, it went from type 1 to type 2 to type 4, so they didn't really have any answers. Sometimes we would be in Boston twice a year, 5 times a year, and once when he had his knee patella had to be tightened on one side and loosened on the other, we were there for a whole month. It wasn't until he started to wear glasses and they were no longer able to help him to see that we went to Mass Eye and Ear. His ophthalmologist then confirmed that he had an eye condition called keratoconus. When his genetic doctor at Boston Children's heard about the keratoconus, she wanted to test him for a gene, and that came back in 2012 that he had the SLC2A10 gene, which was arterial tortuosity syndrome.
Well, my name is Dr. David Warner, and I am an associate professor at the University of Arkansas for Medical Sciences. I became involved with arterial tortuosity syndrome through our genetics clinic. We have an outstanding and somewhat unique clinic here where we see patients with rare diseases, and that's how I became introduced to Aiden and his condition. So Aiden had noticed a steady decline in his vision over a process of a few years, and he was seen by an outside physician who noted that he had increasing myopia and some corneal opacification. And so we wanted to see him to characterize the severity of his condition and see what kind of treatments might be available for him. His vision without glasses was 20/80 in the right eye and 20/50 in the left. And he pinholed to 20/50 in the right and 20/25 in the left. I don't think he's seen this good for years. He pinholed to 20/25, that's almost perfect vision. The first thing we did for Aiden was to establish the severity of his corneal thinning. Because he has keratoglobus and his corneas aren't as strong as other people his age, then his corneas tend to bulge, and that weakness causes increasing myopia. So we did some scans to assess how severe the opacification and the myopia were, and then we talked about a unique and new treatment called cross-linking. We referred Aiden to Dr. William Trattler in Miami, Florida for an evaluation with the hopes that he could receive cross-linking, which would essentially freeze his corneas so that they would not continue to steepen and worsen. These corneas look exactly like they looked 6 months ago before you ever had cross-linking. I can't see one bit of difference. That's a great thing. I don't really consider myself an expert. I'm just trying to learn, and I'm just trying to help Aiden and others like Aiden who really live with some degree of uncertainty. I think one of the greatest things that I can do is to help connect patients and families with resources and information that might be useful to them. And we're happy to continue to support this effort and to try and educate people as much as we can on what ATS does to the eye and how we can help people to keep their vision or improve their vision.
My name is Zolt Urban. I'm an associate professor at the Department of Human Genetics at the University of Pittsburgh. And I study connective tissue diseases, including arterial tortuosity syndrome. Did some measurements of the skin. We look at the thickness of the skin. We look at the resilience of the skin, how elastic it is. and that is seems to be altered in ATS. And the importance of that is that the skin is kind of easily accessible part of the body that reflects what's happening inside the body. So if we see changes in the skin, we can understand more about how, for example, the arteries are changed in arterial tortuosity syndrome.
So in 2014, Aiden has a pneumonia-type illness and ends up with 7-day stay at one hospital, and then we ended up having to meta-flight him back to Arkansas Children's Hospital for care. And that is what started really a twist of fate, because as a parent, you never want to hear, I don't know what ATS is. You never want your child treated just like every other child to fit in this nice, neat little package. Arterial tortuosity syndrome is very unique, and each individual child with ATS will have their own symptoms and react to different procedures in their own special way. So if a physician tries to treat a kid with ATS in the box, they're gonna mess everything up. And so it was very important for me to be able to find a voice and to be able to get information about ATS out to the public, out to other stakeholders, out to other medical facilities.
The ATS clinic came about because of a need for those children to receive multi-specialty care. We learned fairly quickly in the disease that it's not just one organ that's involved. The organs can be the heart, the lungs, the brain, the eyes, the kidneys, and we're still learning about what organs can be involved. And so in order to better care for those children, instead of having them come to the hospital and having to see 8 different doctors in 8 different clinics, we thought it would be best to provide a multi-specialty environment and have all the doctors come to one clinic, provide care for those children at one-stop shopping, so to speak. And so that's what prompted us to do that here at Arkansas Children's Hospital. We had the resources and the facilities to be able to provide a multi-specialty clinic for children with arterial tortuosity syndrome.
It was really a great honor for me to be asked to become the medical director of A Twist of Fate. Because, however, I still haven't seen that many patients, maybe 25 or 30 throughout my whole career, but still I have studied a lot of medical files of patients that I might not have seen in real life, but still it gives you further knowledge about the disease and how you have to manage it, how you have to follow the patients. And so in my position as a medical director, I think it's a privileged position actually to bring that message to patients and to their physicians. Again, we have a clinical team that has a vast experience in the management of patients with connective tissue disorders. And so we have adult cardiologists involved, we have pediatric cardiologists, we have pneumologists, we have dermatologists, we have physiotherapists. So that makes up a great team from the clinic, so we can perform deep phenotyping. On the other hand, we also have a strong research team back in the lab, and so we have Paul and Andy who are supervising a lot of work on the research efforts on ATS. But we also have young people, PhD students, that are committed every day to work with these animal models in order to find out the exact mechanisms, how these molecular defects would lead to a certain disease.
The more samples we have, the more research we can conduct, and it also can account for the variability that's seen in patients. It will be easier for us to make conclusions from the data we have if we have more samples.
This time I started Googling, and then I found some other children that were confirmed to have this gene, which was discovered in 2009. by a Dr. Bert Kellerwerk from Belgium. I could see the children's faces, and I could see my son's face in them when he was younger. I was glad that they had a confirmed diagnosis so early, and I'm just getting it. In 2015, I was on Facebook and I connected with these mothers. I don't even remember how it happened, but it happened, and we were just typing each other, and I met Andrea Taylor, who had started a nonprofit called A Twist of Fate. And that year she was having her first Arterial Tortuosity conference in Arkansas, and she invited us to come.
I guess I didn't really have a perspective of how rare the disease is and how spread out the kids were. So there was really no place that was managing more than one kid or several kids in that way. The Twist of Fate came on, I think I either emailed or called Andrea, and it happened to be that the conference was just starting that day basically. I think I had contacted her on a Friday. And so yeah, then we ended up driving all night to be able to go attend the conference, meet the other families, participate in some research.
But with the nonprofit and all the other mothers behind me, I didn't feel alone anymore. And I could see that Tyrese, once he met those kids at the Arkansas conference, he didn't feel alone anymore either.
It's really amazing to be able to be in the position that I am as the group leader, because I'm not only a parent, but I also become friends with all of these families. And it's really amazing to watch what a unified group that we have. I think that our group is very unique because we really feel like we're family with one another. And so it may be a year in between the times we get to see each other at conferences or group meetings or whatever we do, but it's like our kids, it's completely amazing to watch our kids play together and watch how the families connect with each other, and to watch a new family come into our fold and how they blossom. They seem to be all tight and quiet and, you know, really just reserved. And then after they've been here for an hour or two, you can slowly watch them open up. And after they've talked with one family, and then they might talk with another. And then before you know it, we're crying when it's time to go home because we don't want to leave each other.
So parents may actually be very desperate the moment they learn that their child has arterial tortuosity syndrome. However, if we look now compared to 10 years ago, we already know better what the disease is actually like and what the risks are, and we can follow those patients. We also know that we can do surgery on patients, so we are advancing just by collaborating and putting information of all patients together. And with our further research efforts, I truly think that within the next years there will be new opportunities for therapy to be able to prevent severe arterial issues to happen in these patients. And really that is the most rewarding thing you can think of as a clinician and also as a researcher. Yes, well, that was actually one of the beautiful things of these clinics, to see these patients interacting with each other, interacting with physicians, interacting with researchers. And it's actually, it increases your commitment to the disease, because actually you can put faces on patients, you can put a face on a disease, and so you actually can see what they are coping with during daily life, and that's really motivating and also really rewarding to see that your job is actually important what you do.
And so what A Twist of Fate has done is put us all together. It's allowed us to pool our data, pool our resources. The registry is going to be huge to allow us to follow these children longitudinally over time, putting them all together so that we can see what happens with each of these individual patients. The disease is so rare, the only way we're going to have enough data with enough statistical power to make some scientific recommendations is to put all that data together. And so A Twist of Fate, I think, in my opinion, the most important thing it's done is to bring the doctors and the families and the patients together. And that's going to be huge as we go forward with ATS.
So as a clinician, you're of course very concerned about your patients, and through this collaboration between our clinic, the clinic in the United States and a Twist of Fate, we can gather all this information, we can inform patients about their management, and we can tell them if any complications are actually happening. And we can provide treatment for those complications. So this is actually the most rewarding thing you can think of, both as a researcher, as a clinician.
We are never going to give up. We are going to keep fighting. And it doesn't matter how long the fight has to be. But as long as there is someone in this world that is diagnosed with ATS, we will not stop fighting to try and find a cure.